In our fourth lecture in the biochemistry module, we're going to talk about carbohydrate structure and function. We'll start first with some definitions. Carbohydrates are defined as a molecule that is organized by the number of carbons and functional groups. The common ones to remember are glucose, fructose, sucrose, and galactose. When you name these carbohydrates, it's quite simply taking the aldehydes or ketones within the structure and switching them to aldose or ketose respectively. Simple carbohydrates are noted as obviously smaller molecules, whereas complex ones are noted as longer chains. When you name these carbohydrates, you just have to place the prefix according to the amount. So a monosaccharide is 1, disaccharide 2, poly, many. Another thing we have to talk about when it comes to nomenclature is the D and L form. This form is based largely on glyceraldehyde. D sugars are named as such, sugars with the highest number of chiral carbons with the OH group on the right side of the Fischer projection. L sugars are noted as sugars with the highest number of chiral carbons with the OH group on the left of the Fischer projection. D and L forms of the same sugar are called enantiomers. The D and L forms of glyceraldehyde are listed below. As you can see, the position of the OH group is what's going to dictate this naming. Sugars are often noted as being diastereomers. This is because they are non-superimposable configurations with similar connectivity. Epimers differ at 1 chiral carbon, whereas anomers differ at the anomeric carbon. If you are having difficulty remembering some of these isomerizations, refer back to the second lecture in the organic chemistry module. Oftentimes, you will see carbohydrates in sugars. Anomeric carbons take on an alpha or beta configuration, and because of this, the anomeric carbon is the new chiral center. Alpha anomers have the OH on the anomeric carbon trans to the free methanol group, whereas the beta has it cis to the free methanol group. If you look, the highlighted carbon in the bottom picture is the anomeric carbon. If we want to visually represent carbohydrates, there are two ways we often see it. It's either the Fischer or the Haworth projection. Haworth projections are when they're listed cyclically with the functional groups protruding out at given sides. So at the bottom right is an example of a Haworth projection. A Fischer projection is the backbone, as we often see in organic chemistry. Again, if you're having difficulty visualizing these, refer back to Lecture 2 of the organic chemistry module. Mutorotation is noted as the shifting of cyclic compounds from one anomeric form to another. The straight chain form is the intermediate, so what happens is the cyclic molecule breaks and reforms into a different composition. Monosaccharides are single carbohydrate units. The most common you will see in almost all of organic life is glucose. Deoxy sugars are simply, deoxy, removing a hydroxide and replacing it with a hydrogen. So an example is listed below. As you can see, the 2-oxy group has been removed, thus giving it its name, 2-deoxyglucose, whereas glucose has that hydroxyl group present. There are some reactions that we have to talk about when we go into carbohydrates. The most common is oxidation and reduction. When you take an aldose and make it aldonic acid, or oxidize it, you just simply add a hydroxide. Tollines and Benedict's reagents are often used to detect sugars that are reducing agents. A reaction using Tollines reagent is listed below. Esterification and phosphorylation are also common reactions. Glycoside formations are simply the formation of glycosidic bonds. This is when anomeric carbons link to another sugar. This is the basis for building complex carbohydrates. Speaking of complex carbohydrates, let's go into what they are to finish out our lecture. Complex carbohydrates are noted as when multiple monosaccharides bond together. There are some very common roles that complex carbohydrates have to do in biological systems. Cellulose is what comprises plant structural components, and it's a dietary fiber source for animals. Starch is the main source of energy for plants, whereas glycogen is the animal equivalent. In the next lecture, we're going to go into lipids, a slightly different biological molecule, but nonetheless as important. We'll see you then.